Hi everyone, back to channel. Today we are going to see the Leptospira introgans. So this Leptospira morphological features are, first you can see they are slender, they are spiral and about 6 to 20 millimeter into 0.1 millimeter in size, and they're closely set spirals. You can see they are so closely set and having a hook hands. Here you can see hook hands, and they're actively motile, showing the spinning and bending or sharply flexing movements. So you can see how spinning and flexing. So here you can see tightly coiled spirochetes, hooked ends, seen only in dark field microscopy or silver impregnatation sets. So you can see this is a dark field microscopy. You can see them. So the different species of this Leptospira are Leptospira intrograns and Leptospira biflexa. This intrograns is a potential pathogen of humans, but the biflexa is a saprophyte. So there are also serogroups of this Leptospira introgans. There are 26 serogroups with different serovars, and some of the serogroups are Icterohemorrhagia, Canicola australis, Pyrogenes, Hebdodamis. So here you can see the Leptospira in this with the dark field microscopy. Now let us understand the cultural properties. They are like, it's difficult to culture, so it is not cultured routinely. They are aerobic in nature, that is they require the oxygen, and also microaerophilic. And in semi-solid media they grow up to few millimeters below the surface. So you can see they require oxygen, and the optimum temperature is 25 to 30. If you remember, for most of the species the optimum temperature is 37, but for this the optimum temperature is 25 to 30 degrees, and the pH is 7.2. So what are the media we use? Actually we use the Korthoff's media, Stewart's media, Fletcher's media, Hellingler's, McCollum, Johnson, Harris. All these things can be known as EMJH together. And we also can grow this in chick embryo, especially it grows in the blood of an allantoic vessel, so it's looking so rare. And let us understand the diagnosis of Leptospira if we can't able to culture them. So in the absence of culture we can obtain this by animal inoculation. So in which animal we usually inoculate it? It is done in the intraperitoneally in the guinea pigs. So in the guinea pigs we can do inoculation. And what about this? Here you can see they grow in bacteriologic medium containing the serum, and the nutritional requirement is very high. So resistance than palladium. So this can survive for a weeks in water in wet soil. So whenever some person goes into this, it usually attacks them. So it is mainly coming from the rats. It's present in the rats mainly. So let us see the pathogenesis. These are the zoonotic diseases, that is from the animals to the humans, causing the leptospirosis. So the transmission is mainly transmitted by the man by direct and indirect contact with the water contaminated by the urine and the feces of a carrier animal. Here usually the carrier animal is rats, rodents. So you can see pathogenesis Leptospira introgans is a cause of leptospirosis. So human infection results when leptospiras are ingested or pass through the mucous membrane or our skin. So they circulate in the blood, multiply in various organs. So if you understand, if they once go to the blood, it's easy to go into different organs, producing fever, dysfunction of the liver, kidneys, lungs, central nervous system. So if once goes to blood, it can reach anywhere. So understand how it enters to us. Actually this enters when we have any cuts or wounds, so always be careful when you have wounds, and also at the abrasion of the skin. And it can also by intact of the mucous membrane of our mouth, nose and eyes, especially in conjunctival eyes. And now let us understand clinical features. After that we'll go to diagnosis. So clinical features, it is asymptomatic in some persons, and it is detected by serological tests, and it can cause non-eccentric or eccentric leptospirosis. So what is this? We'll understand what is non-eccentric. Non-eccentric means it just has fever, myalgia, headache, nausea, vomiting, splenomegaly and aseptic meningitis. But eccentric means it presents a hepatic involvement causing jaundice. So whenever liver is involved, then it is known as eccentric. If liver is not involved, then it is known as non-eccentric. So there is also vascular involvement or renal involvement can occur, causing the DIC, disseminated intravascular coagulation or acute renal failure. So how we can detect it in laboratory? Actually, we take specimens like blood, urine or CSF from the affected patient and we collect it. Actually, this Leptospira can die in the acidic urine, so we need to convert this urine into alkaline so that it will not die and we can easily collect it. So how we process the specimens? Mainly by microscopic examination, by fluorescent antibody technique. So what is this antibody technique? The antibodies produced through this Leptospira, if they are produced, we can find it easily. And silver impregnation technique or dark ground microscopy. So these things we do. What about the culture? Actually, this is difficult to culture, so it is not done. We also already discussed. So culture identification is done by the agglutination with type specific sera, and generally it is done by the reference laboratories. So this is the culture properties. And next we have the serology. It is mostly used laboratory diagnosis. We take serum. So antibodies appear at the end of a week. So by week we can identify it, and it is detected by two types of tests. So what are the first one is a screening test or genus specific test, and second one is a specific test or confirmed test. So what are these tests? Let us understand. What is genus specific test? It is where we use the antigens from the non-pathogenic species. So not from pathogenic, from non-pathogenic species they take the antigens. So what are those? You can take CFT, agglutination, indirect immunofluorescence, ELISA or rapid dipstick. So these are the tests. And type specific tests, in this we do the macroscopic and the microscopic agglutination test. So macroscopic is formalized suspension of prevalent Leptospira serovar is used for the macroscopic agglutination of the test sera, and microscopic is the live culture of different serotypes are used, and the agglutination is observed under the low power of the dark field. So let us understand here laboratory diagnosis. It is based on the history of the possible exposure to water or soil contaminated by the L intrograms. So suggestive clinical signs and a marked rise in the IgM antibody mainly. So we are saying antibody mainly, especially the IgM antibody is increased in our body when we are infected with this. So what are the principles and prevention of this? It's avoiding the contact with the contaminated environment, and the treatment is nothing but the choice is penicillin. So treatment of choices, especially penicillin G. We can also use tetracyclines and doxycyclines to prevent this infection. So this is all about very simple topic, that is Leptospira.